My interests have always been on environment and environmental health, whether I was looking at impacts of lifelong exposures to ionizing radiation at Chernobyl in small animals, or in occupational settings, looking at exposure to workers who are working with chemicals and compounds, making rubber and other kinds of rubberized compounds. So it all really centers on environment and environmental health. And I know we're going to have a Meet the Chair event week after this one. And I hope anybody that has time can jump on that, so I can kind of go over what's going on in the department. But we're in a regrowth phase, and are really trying to build ourselves back out into a fuller environmental health science department. And I'm just hoping that we can find ways, at least across the school, to explore collaborative opportunities as well. It's one thing I would say. I think all of us in public health, and probably just about any scientific discipline, collaboration and working with others, with different types of expertise that complement what we're working on is certainly something we do. So we're always looking for opportunities to work with folks. Kind of what I wanted to go over today was use some of the work, some of the NIH funded work we have in Suriname in South America as sort of a case example of this opportunity for collaboration. And then I'd like to go through, obviously, what we're finding in Suriname. It's an ongoing project. It's a longitudinal cohort study. As you can see, this is not anywhere near the exhaustive number of authors, but this is most of the principal authors involved in this study. So we have environmental health scientists such as myself. We have an epidemiologist, two epidemiologists, actually, a reproductive epidemiologist and an environmental epidemiologist. We have folks that have expertise in various clinical parts of what we're doing, especially sample processing and archiving. Because of the kind of exposures we're looking at, we even have folks that are involved in the agricultural industry, because pesticide use is kind of a black box in Suriname. We also have experts in children's neurodevelopment. A pediatric neuropsychologist is part of the team, because one of the principal things we're looking at are impacts to children who are exposed in utero, especially with a focus on neurodevelopment. We have a number of student trainees, and then we also have pediatricians that are involved in this. So with that, I will launch into, hopefully everybody can see this. I'll go over and introduce Suriname itself. Most people, when I tell them I'm working in Suriname, they don't know where it is. They most often think it's somewhere in the South Pacific or the Indonesian region. It's actually a small country on the northern part of South America, but mostly allies itself with the Caribbean countries. So they sort of see themselves as South Caribbean. It's considered an upper middle income country by the World Bank. It's a former Dutch colony, so everyone there generally speaks Dutch, though there are some other languages spoken there. Most people understand and know and can speak English, but there's a number of native tongues as well. So I'll kind of go over Suriname, the scope of our work. My principal focus has been on exposure assessment and biomarkers, and then we'll talk about some of the impacts and implications for health. So Suriname itself, as you can see in this little figure, is a small country. It's actually the smallest country in South America. French Guiana is not a separate country. It's still a French territory. So Suriname is the smallest country. It's about 164,000 square kilometers. Most people live in the capital city of Paramaribo, which is on the coast there, the northern coast. And then another large proportion, if you will, live in the northwest region in a city called Mercury. That's where most of the heavy agriculture is occurring. Their principal agricultural product is rice. And so that's the heavy rice producing region in that area. A lot of pesticide use, I won't spend a lot of time on pesticides today, but it is one of the other chemicals that we are concerned about and that we are currently working on in terms of understanding exposures. The interior region is really Amazonian forest. Most of it is actually quite pristine. There's growing interest in timber in this area. So while it's largely pristine, there is some movement to start harvesting more timber. Most of that's going to Europe and Eastern Asia. It's very sparsely populated. Most of the communities in that area may have 25 to 100 individuals, so very small, primarily tribal peoples, which are formerly called Maroons. They're individuals of African descent, and then indigenous peoples. Even though it's a small country, it's pretty diverse. Population's around 600,000, so could easily fit that population within the greater Birmingham area. There was not a lot of people, but fairly diverse. Hindustani, these are people of Indian descent. Tribal peoples, which are people of African descent, they were formerly called Maroons. And then there are those that identify as Creole. There's a reasonable proportion of people from Java, Javanese people. Most of these different groups came over in different waves following the end of slavery to help with what was at the time a major sugar industry, but they really don't produce much sugar anymore. It's mostly rice in terms of the agricultural product, but I'll touch on that again in a minute. And then there are those that identify as mixed. Indigenous peoples make up about 4% of the population, and then these others represent a very small proportion. Median age is about 30 years. Life expectancy, a little lower than ours, 75 for women, about 70 for men. Mortality rates are a little higher than ours as well, especially some of the infant mortality rates. This gives you kind of an overall view of the population structure, sort of transitioning to more developed profile. The country itself, in terms of diet and disease, is really shifting towards that you see in more developed countries, a shift away from infectious diseases. Not that those are gone by any means, but they're really, the disease burden itself is really shifting towards non-communicable diseases. Obesity rates are actually quite high in Suriname. Access to all kinds of fast foods is quite common. They even have of all things, they have a Popeye's chicken in Suriname. So it's just, first time I saw that, I was like, really? This is what we're exporting? Great. But births per 1,000 women, about 75.9. The reproductive age range starts about 15, 14, especially in some of the indigenous communities. That's just part of their sort of cultural approach. The fertility rate's still pretty high, even though there's very little migration into the country. There's a good bit of migration out. In fact, what you tend to see is sort of a brain drain effect of people leaving Suriname and moving to the Netherlands for educational and career opportunities. Median birth weight is about 3,000 grams, but the percent low birth weight is actually pretty high compared to what you find in the US. And the percentage of women who are having children below the age of 20 is pretty high. On the economic side, formerly bauxite was their big mined commodity, and that's a principal source for aluminum. The company Alcoa had a big stake in Suriname for years, and then a company, government-run company, Suralco, ran the bauxite industry for years, but that's pretty much gone. They really don't mine any more bauxite. Now it's mostly petroleum. In fact, they've just discovered some huge oil and gas reserves off the coast of Guyana and Suriname. And while we should be moving towards using less fossil fuels, there's just a massive inrush of all of the big companies, ExxonMobil, Chevron, Phillips, to tap into that resource. So we're going to see a lot more petroleum extraction going on in and around Suriname in the future. And then there's a heavy focus on gold mining, both large scale, but also small scale gold mining. And one of the things that really got us interested and concerned about Suriname is all of the small scale gold mining. And most of that uses the very old traditional approach that gold mining sort of originated on, in using elemental mercury to amalgamate the gold out of slurries. It causes it to become heavy, it drops out, and then the gold's easy to collect at that point. You just burn off the mercury and you're left with pure gold. And as you can imagine, it's not the cleanest process, so a lot of mercury escapes to the environment. And then even those that are working with it, primarily in the capitals, they process it, it raises a whole nother environmental occupational safety issue, and that most of these people are not working in safe ways with this gold amalgam. There's also a pretty big fish and shrimp industry. Folks along the coast tend to eat marine fish and shrimp. People that live in the interior communities where most of the gold mining is going on, and certainly where most of the mercury pollution and contamination is, rely almost solely on freshwater fish for their protein source. And then they raise bananas, rice, plantains, and then, as I said, there's a growing focus on timber. So our study is the Caribbean Consortium for Research in Environmental and Occupational Health, also collectively called CREO. This particular study is funded by the National Institutes of Health. Fogarty International Center is the principal funder of the work, but there's participation from the National Cancer Institute, the National Institute of Environmental Health Sciences, which is one of the primary institutes that I seek funding from. And that's where the environmental exposure issues really resonated. It's a longitudinal environmental epidemiological cohort study. We've been at this now for about 5 to 6 years with a goal of recruiting 1,100 pregnant women and their children. And the idea was to document exposures that occurred during gestation and early development, and then track these children over as much time as we possibly can to look at the neurodevelopmental and neurological impacts that some of these exposures may have. We hypothesized there would be distinct differences. Certainly in the interior, we felt like mercury exposures would be highest because that's where most of the contamination is. And mercury gets into those biological systems and contaminates fish. It's very similar to what happens anywhere where mercury is a problem in terms of getting into a food source. Nicari, we figured, would be the area where we see most of the agricultural exposure. So while I won't spend a lot of time on pesticides, I will talk about a couple of the elements we're looking at that are key components of pesticides. These are metals that are a key constituent in some of these pesticides that are used there, fungicides, things to control snails. And then in Paramaribo, we felt like the exposures would be fairly low, with the exception that there may be higher lead exposures in Paramaribo, because that's the highly urbanized area. There's a lot of old housing that used lead paint. That's where you find virtually all of the automobile traffic. You really don't find any automobile traffic in the interior. There really are no roads in the interior. So we thought that's probably where we would find the highest lead exposures. They phased out lead and gasoline in 2001. So we thought and still feel like it could be a source of exposure of concern. So we were primarily, and my work was primarily focused on environmental exposures and the consequent impacts on both maternal and child health. We focused on birth outcomes, gestational health in our pregnant women, and then obviously looking at environmental exposures to heavy metals and some toxic non-essential metals, as well as some essential metals. We've been using several screening tools to evaluate neurodevelopment in our children. I've got 4 of them listed here: the Bayley Scales of Infant and Toddler Development. We're using version 3. There's now a version 4 out. The Child Behavior Checklist, which is really what it sounds like. The Bayley Scales is really meant to cover many domains of development in very young children, infants and toddlers. I'll talk a little bit more about that in a minute. The Child Behavior Checklist is really what it's a fairly brief screening tool that parents complete to give you some insights into any kind of behavioral issues their kids may have. We're also using the Child Development Review. And then the other one I'll talk about today a little bit is the modified checklist for autism in toddlers called the MCHAT. And this is used to screen toddlers and young children 16 months to 30 months old for various autism spectral disorder characteristics. So to give you a little bit more information on the Bayley Scales of Infant and Toddler Development, there's several domains that Bayley Scales cover in terms of cognitive development, cognitive ability, language ability, motor skills, fine and gross motor, and then social emotional scales that it's designed to capture. Some of the newer things, the newer version is designed to capture adaptive behaviors, and then listed underneath are some of the details among those. It's not my area of expertise. I've learned some about it. I always feel like I'm needing to learn more. And this is an area that I am trying to develop my understanding of better, so I can communicate better with my neurodevelopmental psychiatrist and psychologist friends. So that's been actually pretty interesting, is to learn about all the different ways that neurodevelopmental experts assess impacts on children or where they are in terms of development. The modified checklist for autism, or MCHAT, has gone through, all of these have obviously gone through several versions. This latest one has a revision to it with a specific follow-up for kids that don't score particularly well. Just so you know, autism prevalence is about 1 to 2 out of 88, so it's a pretty common or commonly diagnosed condition in children, and that metric comes from kids at 8 years old, primarily assessed in the US. It was designed to screen for a few things: impairment in social interaction and communication, presence of repetitive and restricted behaviors and interests, and then it also points to some other developmental delays that aren't specifically autism spectrum disorder. So I won't spend a lot of time on pesticides today. I will spend some time on the metals and then some of the outcomes, but we are particularly concerned about pesticide use. While environmental policies exist in Suriname, at least for some things, there is an absence of policy in certain areas. There's really a lack of enforcement. So they have policies around import and export of different pesticides, but they don't really have enforcement capacity. So they really don't have any analytical ability to test pesticides as they come in, to make sure that what's on the package is what's in the product. We've already found through another study looking at pesticide residues that there are some banned pesticides that are showing up in some of their produce. These are some old organochlorine pesticides, and it could be residual pesticide in the soil and uptake, or it could be legacy pesticides that people still happen to have and they're still using, or it could be that there's some imported pesticides that are, for whatever reason, they contain these banned pesticides. 2 of them that we're particularly interested in because of how heavily they're used are a fungicide called Mancozeb or Maneb. It's a manganese zinc-based pesticide, so we're particularly concerned about manganese exposures. High levels of manganese are implicated in in adverse neurological health. It's heavily used on rice and in the banana plantations. So it's one of the more widely used pesticides, fungicides. And then another one that's tin-based. It has the heavy metal tin as a key component that's used to control snails, especially in the rice industry. So these are two of the most heavily used, and both of them are heavy metal based. And then other metals and elements we're concerned about are both non-essential and essential. We know that mercury is heavily used in the small scale gold mining operations, so we're particularly concerned about higher exposures to mercury. Since lead is a well-known neurotoxicant, you can't really ignore it, even though we do know a great deal about it and mercury in terms of impacts on neurodevelopment. We know surprisingly little about whether or not there's any kind of interaction, or even if they work through independent mechanisms to affect neuronal health, do they exacerbate one another? In other words, is a combined exposure an additive effect, or is it synergistic in the sense that the two together are even worse than either one alone? We were also concerned about cadmium. Cadmium is another heavy metal of concern, and there are some studies showing that in areas where people mine different types of ores for aluminum and things like that, that cadmium can be at high levels. So we were concerned that cadmium exposures might be high as well. Mercury and selenium have kind of an interesting antagonistic relationship. Selenium is an essential element that we really need in very small quantities, but there's growing evidence showing that selenium sufficiency can mitigate some of the negative impacts that mercury can have on neurological health and neuronal health. And then manganese is another interesting one. Again, it's found in the pesticide, but it's also an essential element, and growing evidence shows that high concentrations of manganese can affect neurological health. This is just a quick map, or a small map, showing where our sampling locations are. We have actually expanded this, but these were the major areas that we targeted. Paramaribo is up here along the coast. Nicaria is over here in the agriculture region. And then we did kind of a transect across the country all the way to the far south. And those areas are increasingly more pristine, but surprisingly quite contaminated in mercury. Most of the gold mining occurs in the eastern region, around where sites 3 and 6 are. But we're finding that fish in the far regions, especially out where number 4 is, are actually highly contaminated in mercury. And the thought is that the prevailing winds and atmospheric conditions in Suriname are favorable to a northeast to southwest transport of the mercury. Obviously, being an equatorial region, a lot of tropical rainforest, it's a lot of rain. So we hypothesize that emissions coming from the active gold mining areas don't move very far. They move up into the atmosphere, but then are redeposited through wet deposition and rainfall across the southwest part. And that's what the fish data seems to be showing. So even though there's no gold mining there, they're still getting contaminated with mercury. So we took whole blood samples, which itself, the whole blood sampling and biological sampling has been a learning exercise, especially trying to collect samples in the interior. So many of the areas that we've been going, some of those on the map there are only accessible by airplane. Some are only accessible by boat. So you can imagine the conditions under which we have to collect samples and then try to get them back to the capital for processing. Quite a challenge. All been on pregnant women. First samples usually late first, early second trimester. We also have samples in the third trimester from most of them. What I'll talk about today is the subsample of individuals for which we have the current elemental data for, and that's 400 of our pregnant women. So all of the data I'll show you is blood samples taken late in the first or early in the second trimester for these 400 participants. We analyzed several different metals, non-essential and essential: mercury, lead, cadmium, manganese, tin, and selenium, using inductively coupled plasma mass spectrometry. It's kind of a gold standard way to look for these elements in blood samples, for example, and other biological matrices. Extremely low limits of detection, and obviously subject to very rigorous quality assurance procedures. The laboratory I'm using is one up at the Wisconsin State Laboratory of Hygiene. A colleague of mine that I know has been there for years, analytical chemist. And so they've not only been instrumental in developing some of the procedures to improve these limits of detection, they are also a clinical lab. So many of the procedures they follow are quite standardized. Most of the data was not normally distributed. In fact, it's rare to find environmental exposures such as these, for example, blood metal concentrations, to be normally distributed. So they're usually long, normal, or right skewed. So we used a number of non-parametric and other methods for statistical testing where we needed to, or there were some transformations applied. So into the results. Now the more interesting part of this. So this table really represents the metal concentrations that we have estimated so far. In the first column, well, let me break this table down. So we have various regions in the sample size. Paramaribo, again, being the coastal capital, and the Cary being the coastal northwest city, and then the interior region being those communities in the interior rainforest area, some in proximity to gold mining activities, some quite distant. I put the NHANES values. These are actually the latest ones from NHANES. They've just released an updated set of tables. So these reflect blood concentrations out at about year 2016. The only one that wasn't available was tin. They only have urinary tin in the NHANES, but you can see how our exposures stack up to those in a group of people from the US. This is all data based on women as well. So I didn't combine men in that as well. So first column is cadmium. We had really low levels of cadmium, which was, comforting, reassuring. Most of the cadmium that the general population ends up encountering or is exposed to is through the diet. And then the other large source is through tobacco smoking. And if you look at this value in the bottom here, the 95th percentile for the NHANES, that's probably cigarette smokers. They usually have about 10 times the amount of cadmium in their blood compared to non-smokers. So our women report that they don't smoke. It's very uncommon for women to smoke in Suriname, less so for men. So this biologically sort of reflects that they're not smoking tobacco, and they're very low levels of exposure. Now, mercury and lead were both highest in the interior. We did not expect the higher lead concentrations, though there was some emerging evidence out of French Guiana and in Brazil that some of those interior, especially indigenous communities, had unusually high lead concentrations. We're still trying to figure out what's behind that. It's not the traditional sources, at least that we tend to think of, like lead in paint, or lead in soil, or lead in dust, or lead in water from plumbing. The high levels of mercury we did expect, even though these are really high compared to what you'd find in the US, for example. They're high overall, but fish consumption is quite high in Suriname, both in the coastal areas and in the interior, obviously. Not that there might not be some contamination of mercury moving into the coastal regions, but we don't have a good feel for that yet. The levels in the capital and in the northwest city that are around 2.5 or so median concentrations are not uncommon for heavy fish consuming communities in the US, whether they're along the coast or they're in an interior area where they eat a lot of fish. But the interior concentrations are quite high. Manganese was highest. Manganese and tin were both highest in Neckeri, where those two pesticides, the manganese-based one and the tin-based one, are used most heavily. So we think that might be explaining the difference there. And as you can see, manganese, not terribly different from what you find in the US, perhaps a little higher. Selenium levels are highest in the interior, and then second highest in Neckeri. And those are, these highlighted cells are significantly different at p less than 0.05 for the statistical analysis. And it does seem to track somewhat with mercury concentrations to some degree. And that's something that I, for one, am biologically interested into why it tends to track that way. It actually does that in all kinds of vertebrate species. So typically like the higher trophic level fish that tend to have the higher levels of mercury also tend to have the highest levels of selenium concentration wise. And there's probably some biological mechanism behind that I won't go into, but I think it's particularly interesting that may be the case and may have some implications for interventions. I also put down here the US action levels for both blood mercury and blood lead. The US CDC back in October 2021 just revised their blood lead reference value down from 5 micrograms per deciliter to 3.5. There's still some debate about the blood mercury level, but this is one that's been suggested based on the fact that the fetal compartment tends to concentrate mercury about 1.5 to 2 times what you would find in the maternal compartment. So if you look in cord blood or even some tissues, especially from animals that have been treated, you find that the mercury levels in the fetal compartment are 1.5 to 2 times what you find in the maternal compartment. The main takeaway here, don't get lost in all of this. I put all this up here in case you're interested in dissecting this, but the main takeaways are where we find the highest mercury exposures and some of the characteristics that comprise those groups of individuals that I think make them especially vulnerable. So not surprisingly, the highest exposures are in the interior region. And this is dichotomized on that health action level. They tend to be higher in the older women. That's not completely unsurprising. Mercury does tend to, for whatever reason, accumulate over time and as people age, especially if there's a continuous exposure. And based on all the evidence we have now, these individuals are continuing to eat fish. They really don't have any real good alternative protein sources. Not surprisingly, it's mostly in the indigenous people by ethnicity. Tribal peoples, they comprise the largest proportion of the interior population. And then if you look at folks with the least amount of education over on the right-hand side and the lowest income, they tend to be the ones that have the highest exposure. So it's also an issue of disparity in these communities too. We're revising this table based on the new measures. This is based on the old CDC reference value, but again, same basic pattern. Pregnant women in the interior and their families tend to be the highest exposed, tends to be the indigenous and tribal peoples, again, that comprise those communities. And then again, those with the lowest education and the lowest amount of household annual income are the most impacted. And you know, here they're getting co-exposed to 2 well-known neurodevelopmental toxicants. So when it comes to education status, not only are they not getting education, but they may not even be able to reach their educational potential. So that's another issue that we're really worried about. As we think about this continuum of exposure that starts prenatally and probably continues throughout the life course. With the limited data we have, we should be getting quite a bit more data on this. We have about 600 samples that are being analyzed for all of these metals in the remaining women that we have in our study. So we'll be able to beat this part up. But I was kind of wondering if there were any potential differences among the different self-identified ethnic groups, and there aren't any. All individuals that we have ethnicity data on, even though these numbers are actually very small, I acknowledge that, doesn't seem to be any ethnic difference. So we were kind of wondering if there might be some customs or cultural differences that might explain, for example, the exposures in lead, but doesn't seem to be the case. I'll go through this pretty quick. I just have the one slide for this anyway. Well, two slides, one following this, but we were looking at the relationship between mercury and selenium. This is part of the sort of bench or laboratory-based work that I'm also interested in. Looking at this relationship between selenium and mercury and whether or not selenium can antagonize mercury in terms of its ability to cause neurological effects. This is our entire sample. There does seem to be a modest correlation between mercury and selenium. When I just restricted it to the interior participants only, again, this is a very small number of individuals, but does tend to suggest that there's a pretty good correlation between mercury and selenium. So as individuals on average accumulate or they're exposed to more mercury, at least by way of the way we're assessing it in blood, they also tend to have the higher levels of selenium. So it remains to be seen whether that's actually or potentially protective, because the neurological assessments are ongoing right now. So there's a lot more work to be done there to explore this. But in the absence of being able to just simply get rid of the mercury in the environment, if that were the case, it might provide an interesting way to intervene and perhaps protect some individuals. The mercury use has to stop, but even if it stops today, mercury is going to be in that environmental system for years to come. Okay, on to some neurological data. Again, busy table, so I'll sit on this for a minute. I'll try to stay within the time limit too. We have some data now on several hundred children who are all from the capital region. We haven't been able to collect the data yet and analyze the data. We have the data collected, but we haven't been able to analyze the data yet with respect to the children in the interior. These are children that were assessed using the Bayley scales at age 12 months. And I would think, and the literature suggests, that even though you can use the Bayley scales at 12 months, and it's appropriate to do so, they may not be very sensitive. In other words, to more finer differences in terms of impacts on these various neurodevelopmental domains. Those are across the top here. The cognitive domain, there's two motor domains, fine motor skills, gross motor skills. There's some language domains in terms of receptive language and expressive language, and then a social emotional scale. And what we did find, if you just look at it by sex, and these are female children versus male children, is you do find some significant differences just based on sex. And this is not something that we found unique. It's actually somewhat reassuring in a sense that other studies who've just looked at differences between female and male infants at 12 months old are find the same basic thing. The only thing we didn't find was a significant difference in the cognitive domain, which other studies have suggested. But the differences in motor, fine motor, and the language domains are often found in other studies. And what happens is males tend to receive lower scores in those domains than females. When it comes to lead, we've looked at lead, or the first exploration is looking at lead dichotomized on the 75th percentile. It doesn't look like there's any significant difference based on our initial analysis, categorical analysis, but there is some potential for effects on the receptive domain. There's really not a lot of data out there on the Bayley scales in terms of this kind of exposure at this age. again, as I mentioned, I'm kind of wondering, and I've talked to the neurodevelopmental psychiatrist about that, and she agrees that at this age, you just may not be able to see real differences, which may be actually finer. So this is really more of a cruder or gross analysis at this age. So they'll be followed up again at 36 months. Those are ongoing right now because most of the kids now are out at about 36 months of age. And then we'll see going forward. Hopefully we'll get more funding to continue our assessment over time. Educational level, again, others have found similar effects, that the higher the maternal education, the higher the scores are in these various domains. So you can see that cognitive functioning, fine motor, and receptive language skills tend to improve with education. Again, somewhat reassuring in that other studies have found that as well. We also collected data on maternal depression using mainly self-reported instruments for that. And there's some indication that as depression screening indices increase, you see a decrease in some of the cognitive domains areas, particularly motor skills in children as mothers' depressive symptoms go up. And then we looked at sort of the correlation between lead and Bayley's just to see if there were negative relationships. Seems like there's some indication that there is some negative trends. Only one of these was significant at 0.05. That was the one with receptive language that also sort of bordered up here with lead levels. We also looked at the MCHAT. In other words, this is the tool that's used to assess or screen for autism spectral disorder characteristics. We looked at both lead and mercury. Right now, we're also looking at mercury in the children in the capital with respect to Bayley scales. I don't have that data to show you on the previous slide, but in talking to the sub-team that's working on that, it doesn't look like there's any association with mercury. But again, the mercury exposures are pretty low in the capital region, at least the maternal exposures. So just to remind you again, kind of where we are with this, we have high lead, high mercury in the interior. Again, I have the NHANES as kind of a reference. And this includes children from across these areas, though there's fairly low representation right now from the interior for the MCHAT. This was collected at 16 to 30 months of age. So fairly recently, we collected some of the data on the interior, and we don't have all that in yet. But this is the first analysis of about 500 kids. And you'll see that we're not really seeing anything when it comes to lead, especially as we dichotomize on using that blood concentration reference value or mercury. Here are the p-values. I used the Fisher's exact test for this, just a two-tailed test. If you use a one-tailed test, which would be fair to do, in my opinion, with something like mercury, where we know it has a neurodevelopmental effect, this would then become significant at p less than 0.041. But I'm holding out on worrying too much about that until we get more data and more moms with exposures into the study. We didn't see any effect of low birth weight. We didn't see any effect with preterm birth. Those are here. I thought we might see something with sex, but we didn't. Males tend to be diagnosed much more often than females, about 1 to 5, female to male when it comes to ASD diagnoses, but these aren't diagnoses per se. These are basically just results from a screening tool for which you would then use to identify individuals that you would want to do a more comprehensive diagnosis on. So that may be part of it. It did seem like we found a high number of individuals at high risk. And I think we need to explore that a little further because, as I mentioned, it generally affects at least children out at 8 years of age. Autism spectrum disorder diagnosis is about 1 out of 40 or so. So I'm not sure what's going on here, but that seems awfully high to me. Then we didn't see any influence of education. And we also looked at the influence of BMI, and we didn't see any influence there. So far, we're not seeing any strong relationship with the environmental exposures, but with more data, especially with mercury, which most of the mercury that we've analyzed is methyl mercury, which is the real neurotoxic form. We may end up seeing something there. And then I guess I should bring up the fact that those individuals in the interior also have the highest concentrations of selenium. So it may actually be playing a protective role. There's some really great sources of selenium in the interior region and in Suriname that people do include in their diet. One of those is fish. We've analyzed these fish for both fatty acids, including omega-3s, as well as selenium, and both the freshwater and marine fish from Suriname are excellent sources of selenium. And then they also grow a lot of Brazil nuts in Suriname, which are the one food item that's probably has the highest concentrations of selenium. In fact, so much selenium that if you eat too many of them, it could be toxic. So you got to be careful. All right, so wrapping up. Not much research really has been done in Suriname. There have been a few Dutch researchers that have done some work. There has been very little in terms of environmental health research in Suriname. So a major gap that we're trying to fill with our work. The neurotoxic metal concentrations and exposures are highest in the interior, at least based on what we've seen so far. There's a number of other factors that I think can contribute to the vulnerability of those populations in the interior. We're trying to source exposures because even though we know the mercury is coming primarily from the fish, we still get pushback, especially from those in the gold mining industry about is it really just coming from mercury that's used in the gold mining? I would say yes, but we're actually using some stable isotope chemistry, looking at stable isotopes of lead to try to pinpoint that and identify a signature. We're doing the same thing with lead and some environmental samples we've collected, both food items, cookware, soil, and they actually do collect some small animals for consumption, small deer and those kinds of things that they harvest with lead shot. So we're looking at stable isotopes in lead as well to try to identify those sources. We're going to continue to track the children and the neurodevelopmental outcomes. We'd like to extend that into looking at older adults who have had a lifelong exposure to all of these, to these heavy metals especially. And what we'd like to do is obviously get to the point where we can look at these as a complex mixture in terms of biostatistical analysis, as well as explore interactions between some of these elements. Right now, we just don't have the power or the data for that. Here's a number of the folks that are involved, acknowledging our funding source. Like I said, hopefully we can keep our funding going. That's kind of what keeps me up at night and wakes me up in the morning, is trying to keep our folks in Suriname funded and keep ourselves funded. It's been a really interesting and concerning study to be a part of. At the same time, I've made some great friends in Suriname, and it's just been a joy to work there. So anybody interested in talking about international research, I'd love to visit with you some more. I've learned a lot, and I'm always learning from others.